Good morning, everyone. Today I'll be discussing a recent study we've been working on that focuses on hominin-carnivore interactions at Escoural Cave in Portugal. The study is based on combining traditional zooarchaeological and taphonomic methods with quantitative approaches, including multivariate statistics and machine learning. And the aim is to identify the main agent of accumulation of a rabbit assemblage we found at the site recently, and discuss possible implications for Neanderthal subsistence and cave use. So Escoural was discovered in 1963 during mining activities for extracting marble. It is a karstic system with a main gallery of around 15 meters connected to other channels. We think that the original entrance, which would have been next to the highlighted area in Figure B, collapsed after Mid-Paleolithic occupations, but investigations are ongoing to determine exactly when that happened. In the 1990s, excavations unearthed Chalcolithic occupations outside the cave, Neolithic burials, and Upper Paleolithic engravings inside the cave, as well as Middle Paleolithic remains. And preliminary analysis of these lithic industries indicate a predominantly expedient technology made mostly on quartz. And the macrofaunal analysis that were carried out back then suggested that carnivores, primarily hyenas, were the ones responsible for most of the faunal accumulation. So in 2020, the Finisterre team returned to the cave to clean a few profiles and take samples for dating and micromorphology to understand site formation better. And what we know to date is that the sequence that contains Paleolithic remains inside the cave comprises differentiated layers with clear depositional episodes and low energy environment. And there was only one data available from the 1990s that established an age of 48.9 thousand years through uran and thorium dating. However, we now have two new radiocarbon dates from bone remains that have placed levels 2 and 3 within the Upper Paleolithic, while level 4 remains ambiguous. Unfortunately, a rabbit assemblage comes from layers 2 and 4, which could mean that some of the sample comes from the Middle Paleolithic. However, since the dates are inversed, this suggests that the layers may have been subjected to mixing processes, some of the materials at least. So there is some degree of reorganization of the materials, and so we need to carry out further dating to see what levels we have of Middle Paleolithic occupations exactly. But we will know more about this soon. So when the study was carried out, we only had one data available around 50K, so we assumed an association with Neanderthal. So our sample of rabbit remains includes over 1,300 bone fragments, which we analyzed following established zooarchaeological and taphonomic protocols for rabbit assemblages. We explored skeletal representation, we quantified NISP, M&E, M&I, percentage Mao, and percentage of relative abundance. We determined age using epiphyseal fusion stages and tested whether density-mediated attrition could have biased skeletal representation. Regarding taphonomy, we explored breakage patterns and recorded important features we observed on green fractures, identified cylinders, notches, and helical fractures, recorded carnivore damage in the form of pits, punctures, scores, and crenulated edges, as well as anthropogenic marks and other post-depositional modifications. And additionally, we carried out some statistical and machine learning analysis to classify the Escoural data. We performed a PCA analysis and then used 3 ML mod algorithms, random forest, support vector machines, and gradient boosting machines on skeletal abundance data. And on the other hand, we use hierarchical clustering on principal components and k-means cluster analysis on the taphonomic data. These quantitative approaches can detect patterns in complex data that we wouldn't be able to detect otherwise, and therefore enhance the reliability of the classifications we make. So in order to do this, we need a data set of reference assemblages, data with which our Escoural sample can be compared. So we did a search in the literature of both archaeological and naturalistic rabbit assemblages reporting taphonomic data and found 52 assemblages with skeletal abundance data and 34 with taphonomic data. These sample sizes are relatively small, so we pre-process the data using methods like imputation, feature selection, bootstrapping, and internal cross-validation to then subsequently train the models with hyperparameter tuning. Later, we assess the model's performance using precision, recall, and the F1 score. And of course, machine learning usually works better with larger data sets. However, with smaller data sets, it can still be useful when the data sets are balanced and when these pre-processing techniques are done. So moving on to the results, our MNI was 19 with a minimum of 3 infantile, 6 juveniles, and 10 adults represented. The highest represented elements were calcaneae and metatarsals, followed by tibia, pelvis, and cranial elements. Something else we can see from the skeletal profile is that hind limbs are better represented than front limbs, and that axial remains are virtually absent. The results of the Spearman's correlation suggested, however, that the lack of axial elements is not related to density-mediated attrition, since the correlation with bone's density values was non-significant. And when we look at the results of the PCA of skeletal abundance data, we see that there's a high variability and overlap between the predator groups and samples in general. And the Escoural sample falls near the human cluster while also overlapping with other carnivore groups. So this may suggest that either a complex taphonomic history at Escoural, or alternatively that the PCA does not fully capture the differences between these highly variable predator patterns. The 3 machine learning models performed differently. So whereas the random forest and the support vector machines yielded moderate accuracy values, suggesting classifications may not be very reliable, the gradient boosting machine performed much better, yielding an accuracy of almost 80%. And so if we trust this model's classification, the Escoural rabbit assemblage was very likely accumulated by terrestrial carnivores, a group that would include lynx, fox, wildcats, etc. Bone surface preservation was generally very good. We counted 8% of bones bearing tooth marks, which were more frequent in pelvis and ulna. Digestion damage was highest in tarsals and ulnae. However, no extreme digestion traces were documented. Overall, this degree of damage also fits very well with the taphonomic signature left by lynx. And we didn't find any cut marks and only 2 burnt remains, which could be post-depositional. The comparison of tooth mark sizes are a bit ambiguous, but they also suggest that the rabbits were consumed by small terrestrial carnivores, as the Escoural sample overlaps with that of foxes, wildcats, and lynx cubs. When we look at all the taphonomic variables together, including the percentage of cut marks, percentage of burnt remains, tooth marks, adults, digested remains, tubes, and complete lung bones, under the lens of the hierarchical cluster analysis, we get the following result. There is an overall alignment with the known predator groups and a clear distinction between human-made assemblages and the carnivore-made assemblages along the first dimension, with the human clusters on the left. And Escoural falls near to other terrestrial carnivore assemblages also in this analysis. The results of the K-means cluster analysis was slightly more reliable, and it also showed similar results. So we have cluster 2 and 4 containing, yes, thank you. Clusters 2 and 4 contain assemblages with human inputs, clusters 3 and 5 a mix of other carnivore assemblages, and Escoural clearly shows taphonomic similarities with Cova del Gigante 3A and Valmaillo, both of which have been interpreted as lynx-made assemblages. In conclusion, the rabbit assemblage from Escoural likely resulted from the activity and accumulation of a mixture of terrestrial carnivores, among which the Iberian lynx stands out. Multiple agents may have contributed to the assemblage, but it seems clear that humans were not involved. These results highlight the potential of statistical and machine learning techniques to enhance taphonomic analysis and distinguish between accumulating agents in very variable data sets. Most of what I've presented it has recently been published in this paper you see on the slide, which you can access for more details and also to access the data sets and the code we used. And in the future, we will continue to explore through additional zoological and taphonomic analysis the macrofaunal remains of the old collections and new excavations, and test whether the cave was mainly functioning as a carnivore den that was only occupied by Neanderthals intermittently, or whether Neanderthals focused on different or similar prey as the carnivores represented in the assemblage. This study was carried out within the Finisterre ERC project and the Marie Curie Project BITES. I want to thank the co-authors of this study, Ana Rufà and João Cascalheira, the excavation crew at Escoural, the organizers of this wonderful conference, and all of you for your attention. Thank you.